So we'll now move on to our next speaker, who is Jeff Milton. He is the founder and CEO of Jolla Labs. And Jeff will talk to us about how artificial intelligence will drive rare disease therapeutics. This is one of my interests. So hello, Jeff. Welcome to the meeting. And
hi.
Looking forward to your talk.
Great. Thanks for inviting me. And let me go ahead and bring up my slides here. I feel like this is going to be a very good collaborative presentation with the previous speakers, especially Winston, because it's really a story about data. And if you don't know anything about artificial intelligence is really the foundation is rooted in having data. And so you'll kind of see that theme here. Can you all see my slides okay?
Yes.
Okay, great. And I'm not cutting out, right? Everybody can hear me. Okay. All right, so let's talk about the drug discovery and development process. This is typically a very long process. And I like the analogy that Winston gave about climbing El Cap. The idea that you can study each area and understand all the treacherous parts of all and divide up this into compartments that can be studied and interpreted in a way that becomes somewhat reflexive later on is a very powerful analogy. And that's something that AI can actually help to do. And so when we talk about the path for what it takes to create a drug, this is typically what people quote. It's 10 to 20 years. There's a huge exploratory research part where it's understanding the disease itself and the target. And then there's the discovery or finding the drug, preclinical development, and then the clinical development. And this is obviously a very expensive thing. This is anything that's sort of a standard drug that goes into the clinic these days. 90% of them fail in the from phase 1 to approval. And looking at this chart here on the right-hand side, you'll see that the new drug entities are the lowest, and then the biologics are around 10% success, and then those are the repurposed indications. And most of the time, these fail because of lack of clinical efficacy and toxicity. And so those are obviously the two areas that you want to focus on as far as being able to optimize the process. Now, for the sake of what we're talking about today, a lot of this is discussing RNA targeting modalities. And so there's three basic approaches to this. There's the RNA-seq mediated ASO, there's the steric blocking, splicing ASO, and then there's the siRNA approach. And so the great thing about this is that, and this was shown earlier, that because this mechanistically is very well characterized, it's a very rational approach to drug discovery, and the fact that the chemistry, the chemical modifications typically don't change as much. It's the sequence that changes, which makes this something that's really amenable to the AI approach. If the chemical modifications change, then you're talking about a degree that's very hard to nail down. But from the point of view of sequence, those are variables that we can manage. And so that's something that's really amenable to the machine learning AI approach. And so we've talked about this before in this session, and it's definitely one of my favorite stories as well as far as a drug. But I'm going to go through a little bit different approach and just kind of show you some of the history here of how this was created. And so if you look at the timeline, this is a 20-year drug from the point of view of understanding the targets, understanding the mechanism of action that was the goal as far as alleviating SMA. And then there's a screening process, which was probably around 2 or 3 years. And it was a collaboration between Cold Spring Harbor and Ionis Pharmaceuticals. 2006, the ASO was found. then there's the preclinical study, and then there's the clinical. And so you can see there's a lot of work that went into this. This is many, many, many millions of dollars and a lot of time. And so the idea that we're going to compress this down and make this a very short process for ultra rare disease and rare disease in general is somewhat daunting when you look at this. However, again, looking back at LCAP, here's our first explorers, and we can learn from this, understand it, and make it and optimize it. So the idea here is that we want to be able to take a screen, find candidates, and jump to approval. That sounds great in practice, but that's not the reality of the situation. But that's kind of what we want to do. We want to kind of have that in our mind as we're designing a drug that's going to succeed from the start. And that's, but we can break this into chunks that will allow us to make those leaps of faith in a sense. So one of the main areas that is really kind of not really solved, but there are definitely tools out there for predicting toxic compounds. Now, these are in vivo tox, and they don't necessarily translate to the clinic, but there's definitely an approach where you can take sequences and a particular modality, whether it's a splicing oligo or an RNA-mediated ASO, and if you have enough data, then rodent tox data, then you can make a machine learning algorithm that will predict at design time some sort of probability that it's going to be either liver toxic or neurotoxic. And there are only and it's very specific to the chemistry. So it's basically varying the sequence. So what you're doing is essentially taking the sequence with a particular modality, a full mo splicing oligo, and the genomic context of that, and then you're able to predict. And with the enough rodent tox data, you can make some algorithms that are actually pretty good. And this has been done before. And so this is one thing that's actually a nice first step toward predicting or for using AI. The great thing about this is that if you can predict further down the pipeline, or predict if an ASO is going to fail in late stage preclinical studies, that's the best case scenario because you're not wasting your time and having to go back and revisit the screening process. So the more you can push your predictability down into the later stage development process, the better. So this would be my first approach toward developing a prediction algorithm. And there are all sorts of bins that we can put toxicity into. Hepatotoxicity is one of the major focuses for ASOs, carcinogenicity and genotoxicity and cardiotoxicity. These have biomarkers, and these are measured in structured ways. And I'll talk a little bit about structure in a little bit, but these are things that can be captured very efficiently. And then what you can do is actually load them into, and for example, we have a design tool that allows us to load our models into and actually lay out, in this particular case, it's an RNA-mediated knockdown around a gain-of-function SNP, and we can actually try to predict or use these tools to help us predict liver toxicity. And so then we would weed those out at the design time. And so there are all sorts of parameters that you can get, and these clinical analyzers are very good at spitting out very structured data. However, getting them hooked up and connected to a database is another leap that you have to go into. So the infrastructure is very important to be able to capture this stuff in an efficient way. And that leads to what I'm going to talk about next, which is really what I call the shape of the data. And the data, how that shape of that data is super important to be able to build these tools. And what I'm going to do is actually jump into a story that goes way back, and I'm going to talk about scurvy a little bit. And just digress. I'll come back to SMA. But I want to talk about scurvy in the context of data. And, this is a disease that estimated to kill 2 million sailors when basically at a time period where genetically we were not prepared to go on these large journeys or large voyages across oceans with minimal dietary constraints. And so these, we don't, unlike a lot of animals, we don't produce our own vitamin C, so we suffered scurvy as a result of that. But what's interesting about that is that as early as I actually have a copy of Vasco da Gama's journal, because it's actually one of the few, I think one of the only, that is first-person account of the voyage to establish a route around the Cape of Good Horn and into India. And he actually says in 1490 in this journal, the Captain Major, which is Vasco da Gama, sent a man ashore, the messengers with instructions to bring off a supply of oranges, which were much desired by our sick. So Vasco da Gama actually lost several hundred, or I think about 130 people on his journey to scurvy. But he knew for some, and the fact that citrus was mentioned a couple times in the journal, but there was a part here where he actually made a specific reference to oranges. It was also referenced by James Lancaster, who was by surgeons and by other, I guess, scientists at the time, or physicians at the time, who understood that oranges or citrus were important for addressing scurvy. But and then of course, the first real clinical trial, a sort of documented clinical trial, was run by James Lind, the Royal Navy, ran a clinical trial on a voyage, and he rediscovered old data from John Woodall and the East India Company actually using citrus to treat scurvy, and published it he published this in 1747, saying, that, but actually only dedicated a little bit of a section to actually saying that scurvy was alleviated by oranges. And it wasn't until 1795 that the English Navy actually introduced fresh oranges and lemons into the diet. So think about that. That's 50 years after Lind had actually published this work saying that it was useful. And that's because there was a lack of communication infrastructure, there's a lack of integrity, and there was a lot of misinformation. And people who had political power could actually push forward their information versus actually having real raw data. And so this is super important. You could see how long it took, several hundred years, to actually come up with a remedy that was established, because that infrastructure wasn't there. And so it led the doors were open for interpretation, and so that continued for several hundred years. And so when we look at infrastructure for what we've got as far as developing the drugs that we're talking about, the screening part of that infrastructure is typically the most robust. So there's the primary screen, there's a dose response, and these are this hunting for the ASO that actually does what you want it to do is actually typically an automated process, and it's very robust. It's got and you're talking about structures that are not changing as far as chemically, but they're changing in terms of sequence. And so I like to think of this, it's ripe for AI, and the pun is intended there. This is actually the process where AI should be a part of all screening, because ultimately it is it's actually the low-hanging fruit. I actually didn't intend that pun, but in any case, I'm going to jump back into SMA, and I'm going to talk about the screen that they did for SMN. And what you can see here is this is actually a presentation that's on YouTube, and I urge you to look this up. This is a great presentation by Adrian Craner, and he talks about the actual screening process. He talks about the whole development, but specifically here is the actual screen. And there's a total of over 500 ASOs, and the yellow part here was the part that was done by Cold Spring Harbor, and the rest were done by Ionis. And what you can see, and if you were to synthesize these, just to kind of put this in the context, it's a very expensive. Just to synthesize, it's about $400,000. Well, at the 1 micromole, you probably would use less than that. But in any case, Spinraza falls out here, but if you were to apply some models to this, and this is some of the models that we've developed at La Jolla Labs, you could take this screen and actually provide a map that sort of gives you a guide for where you're actually going to apply these ASOs. So you wouldn't apply them to the parts that are going up in this particular area, because you want to block the exclusion. And so in this particular case, you would eliminate essentially most of the ASOs. Now, this isn't drawn to scale, so this is an exact representation of it, but it kind of gives you an idea qualitatively of what you can get from using a machine learning approach to actually designing your ASOs. And we're able to recapitulate what SMN does. And so in this particular case, and we're using a lot of the ECLIP data and the RNA binding protein data that's out there. And so we're training on this and actually learning where is the best place to put an ASO. And so because everything is so structured, because we have a structured molecule, and we have a structured representation of the molecule, it's not changing chemistry, it's changing sequence, and we know the genomic context of it, and we have very structured sequencing data that we're training on. That allows us to do this. And so what we can do is we can kind of look at where the landscape is right now for what sort of things we can train on. And really what we want to do is we want to bridge these lakes of variables, essentially. We want to put bridges across them to say, we want to do in silico to in vivo toxicity. We want to do in silico to activity in vitro. We want to do in vitro to in vivo activity, because that's not exactly a solved science as well. And we want to go in silico to tissue distribution. And this kind of gives us the ability to learn all these little sections in a way that allow us to have better predictability going down into the later stage of the pipeline, and ultimately get to the point where we can say, okay, we can do everything mostly in silico, maybe some in vitro work, and have a really good chance that we're going to be successful in the clinic. And that's the dream, right? But that's not where we are. So where are we today? Well, we know there are people who are doing in vivo toxicity successfully. And we at La Jolla Labs are doing in vitro activity. And we know there are people who are doing in vitro, and we're doing that as well, in vitro to in vivo activity. And so these are where things are right now. That by no means has this this discipline in a state where it can predict anything clinically at this point. There's absolutely there are just too many hurdles at this particular point. So that's definitely, this is really the representation of where we are. And so the idea here is that we're going from research to clinic, but we could also go back from clinic to research. And this is where we bring in things we need, especially when it comes to rare disease. We need to bring in things like natural history studies, clinical trials, data. So things like adverse events that we talked about earlier, and injection site reactions, all that sort of stuff. If there were an open space or a pre-competitive space that allowed us to say, okay, this particular, because some of these things are very sequence specific, the chemistry may not have changed, but it's sequence specific, or that's something that we can train on. And so that sort of this going back has to happen in order for this dream to happen, as far as being able to use computation to predict the success of a candidate. And so there are all sorts of tools that allow us to do this. There's a guidance for natural history studies, and there are all sorts of technologies that are out there that are allowing us to capture these data, and especially for these neuro indications where there are specific cognitive tests you can do, and there are even EKGs that can be used to capture data, and that data can then be used to build really nice, sophisticated control studies that allow us to actually push a particular candidate down through in a clinical trial faster if it has a natural history study associated with it. And so, just to give another example, if you can do things like video analysis of motion, and capturing motion over the period of time and being able to train on that data set, which then allows us to give a baseline, which then allows us to determine in the external clinical studies whether or not we're deviating or the efficacy is improving the actual clinical study faster. And I just wanted to highlight just very quickly that, the FDA does fund natural histories studies, and these are the 2022 awards. And so I would urge anyone who's interested in doing this to pursue these things and get this out there. And to kind of wrap things up, following up on what Winston was talking about, we have a desperate need for a pre-competitive data space. Essentially, that's screening data, tox data, clinical data. All this stuff needs to come into place. In the rare disease community, it has to happen in the rare disease community, because working in silos is just not going to be sustainable. And in order for rare disease therapies to progress, we have to work together, and everyone has to work together to create this space to make this happen. And one of the things I want to say is that, what we're going to do, we have a website called genome.bio. This is right now it's a blog, and it is a blog that sort of talks about some of the rare disease things that we're working on. But we're going to expand this out, where we're going to supply some screening data that we have, and we're also going to supply some tools. So we're going to provide some of the software that we have to allow you to do your own designs. And we're calling that the sandbox. So it's our design sandbox. And so with that, that's all I've got. And I just want to thank Nyanwei for inviting me. And I'm really excited to be a part of this awesome group.